My name is Dr. Christopher James Ward from the Kansas University Medical Center, and today I want to talk about the use of exosomal polycystin-1 level to diagnose and monitor autosomal dominant polycystic kidney disease. I'd like to personally thank the organizers of the PKD Connect conference for allowing me to present by Zoom. I have a few home issues at the moment, and I need to be around for my sick wife. So here's the standard activity disclaimer for the PKD Con, that we're not responsible for any clinical decisions made based on the presentations today. And here are my disclosures. I have an R01 from the NIH, NIDDK. The PKD Foundation gave me funding to develop this assay that I'm presenting today, and as did Regulus Therapeutics. And I had a consultancy from OrbiMed for $1,500, just asking about exosome biology. And this describes my biography. I trained as a medical doctor from 1981 to 86 in Edinburgh, Scotland. Did my Ph.D. in Birmingham University in England, UK, from 86 to 1990 on neutrophil elastase. And since then, I've worked on AD-PKD, first in the Institute of Molecular Medicine in Oxford, then at the Mayo Clinic, and then in 2013 I branched off on my own, and I've been working in the KUMC ever since. I was involved in the cloning of the PKD1, the TSC2, and the PKHD1 genes, but I'm probably best known for reagent development and for the exosome hypothesis of AD-PKD. At the moment, I'm developing assays for the diagnosis and prognosis of autosomal dominant polycystic kidney disease. So this is what we're looking at. This is autosomal dominant polycystic kidney disease. This kidney is about 10 times bigger by volume than a normal kidney. And of course it's autosomal dominant, so that if you have it, 50% of your children will have it. It's a very unfortunate disease, and we want to relegate this phenotype to the history books. So we're going to talk about the products of the three polycystic kidney disease genes: Polycystin-1, Polycystin-2, and fibrocystin, the products of PKD1, PKD2, and PKHD1. Now the first one I'm going to talk about is Polycystin-1, which is a very large protein of 4,302 amino acids, and it spans the membrane 11 times. So these are transmembrane spans. As a huge ectodomain, mainly made of PKD domains here, PKD repeats. But what we're going to concentrate on today are the monoclonal antibodies that I've generated to these proteins. And the first one is 7E12, which is to the leucine-rich repeat of Polycystin-1. It's a very good antibody, and we know that it's specific. Now this is Polycystin-2, which is a calcium channel subunit. In fact, it's part of the TRP family. In fact, this is TRP-P2. It's 968 amino acids in size, and it spans the membrane. It spans the membrane 6 times here. Now we've got 3 monoclonal antibodies that are specific to this. 2 are commercially available: YCE2, DE3. And this is the inaugural description of a new monoclonal I've made to the top domain called CJW9D5, and it's IG2B, and we'll be using this in some of the work I'm about to show you. So this is fibrocystin, the product of the PKHD1 gene, which is responsible for autosomal recessive polycystic kidney disease. The protein is 4,074 amino acids long, and it has a cleavage site, a protein convertase cleavage site at 3617. Now we've got monoclonal antibodies to this large type 1 membrane protein, and they bind at the N-terminus between amino acids 143 and 238. So that's in the first 3 TIG domains. So there's another little protein that we've got to discuss, and it's called CU62, and it's a small protein that binds to the PKD repeats of Polycysin-1. Now, it's a small protein. It has a signal peptide here, and it has 5 glycosylation sites, but it doesn't have a transmembrane domain. Its unglycosylated mass is 24 kilodaltons, but when it's glycosylated, it goes up to 42. And it can interact with itself, and it can interact with any of the PKD1 repeats of Polycysin-1, but especially repeats 15, 16, and 17. Okay, so the big question is, where are these proteins? And we found that the biggest source of Polycysin-1 and fibrocystin is in urinary exosome-like vesicles. So we also secrete 100 nanometer diameter vesicles into our urine, and they're a very rich source of Polycysin-1, as you can see here. And this is deglycosylated, Polycysin-2, and fibrocystin. And as you can see, fibrocystin is very large. So here we've resolved PKD exosome-like vesicles on a 3 to 30% sucrose gradient in heavy water. So we put the exosomes at the bottom of the gradient and allowed them to float up while we centrifuged at 200,000 G. And as you can see, the Polycysin 1 and the Polycysin 2 and the fibrocystin and the CD133 and the CU62 are all resolving in the same fractions, whereas ACE2, angiotensin converting enzyme 2, the receptor for COVID, is actually on a different set of exosomes. So I've always been interested in... Hi, my name is Dr. Christopher James Ward from the Kansas University Medical Center, and today I want to talk about the use of exosomal Polycysin-1 level to diagnose and monitor autosomal dominant polycystic kidney disease. I'd like to personally thank the organizers of the PKD Connect. The receptor for COVID is actually on a different set of exosomes. So I've always been interested in proteins that might interact with Polycysin-1, and one strategy I used to try and find these was to use label-free proteomics on individuals that had PKD1 mutations. The idea was that if you had a PKD1 mutation, then the amount of Polycysin-1 in your urinary exosomes should be decreased, and with it, anything that's binding to the Polycysin-1. And so what we did was we looked at 13 individuals with PKD1 mutations, 18 controls, age-matched, and we looked at 2008 proteins using label-free proteomics. This is a volcano plot. In this direction, a protein is decreased in PKD1 versus control. And up in this direction, this is the log 10 adjusted p-value, and it's Bonferroni adjusted, okay? So 3 is 1 in 1,000, or 0.001 p-value, okay? So what you see is that PKD1 is decreased, polycystin's decreased, but this little fella here, CU62, is really decreased in polycystic kidney disease exosomes. And so we followed this one up. So these are primary cells isolated from a human kidney and just grown for 1 or 2 passages. I get them from Darren Wallace. These cells have been polarized, and they have primary cilia, so they're serum starved and polarized. And the blue is the primary cilium. The green is either Polycysin-1 here or Polycysin-2 here, and here you can see CU62. I've created 2 monoclonals to CU62, and they both work well on Western and by IF. But in migrating cells, you have a slightly different picture. Here we have a cell that's migrating in this direction, and it's leaving behind a trail of retraction fibers that are intensely polycystin-1 positive. They're also tetraspanin 4 positive. Now, tetraspanin 4 is a marker for the retraction fiber, but it's also a marker for a new type of extracellular vesicle called a migrosome. Okay, and as you can see here, you've got complete co-localization between the two. What's interesting is that the cells that are making these retraction fibers are actually megalin positive. Okay, and here are the retraction fibers associated with them here. And these cells are actually proximal tubule cells. So here's a close-up of some migrating cells. Now, I've stained a cell with Floyd in here, okay, just to give the outline of the cell. As you can see, the retraction fibers are intensely positive for polycystin-1, but you have these vesicles on the retraction fibers that are intensely positive for Cu62, and the Cu62 seems to be forming a coat over the polycystin-1 on these extracellular vesicles. So these extracellular vesicles are classified as migrosomes. To talk about here, and that is focal adhesions. Now, Pat Wilson, many years ago, thought that polycystin-1 co-localized on the focal adhesions, and on proximal tubule cells, she's right. Polycystin-1 here co-localizes very tightly with talin and with alpha-actinin here. And also PC1 and Cu62 are together on the focal adhesion. Fibrocystin and Cu62 and polycystin-2 and Cu62 also are in close proximity on the focal adhesions. And so basically what I'm saying was Pat was right. The polycystins are on the focal adhesion. So let's return to the main theme of the talk, and that's an assay for polycystic kidney disease. And here we have a Western blot that's looking at the levels of polycystin-1 here versus CD133. And so it's the ratio of polycystin-1 to CD133 that we'll be assaying here. Now, there's a linear dose response for this assay, but the in-batch CV is about 12%. The between batch coefficient of variation, or CV, is about 20 to 30%. Having said that, we can still use the assay to distinguish between individuals that have got PKD1 mutations and people that don't. And so we looked at 22 wild-type individuals and 15 people with PKD1. And in collaboration with Regulus, we showed that we can easily distinguish people that have got PKD mutations from those that are wild-type here. And the p-value is astronomically small here. So we were able to show that the PC1 CD133 ratio correlated inversely with height-adjusted total kidney volume. As you can see, as the PC CD133 ratio goes up, the total kidney volume drops, okay, implying that the more polycystin you've got, the less severe your disease. We got a p-value of 0.014 and an r-squared of 0.63. So what do we want to do in the future? We want to improve the Western assay for PC1 CD133, improve the in and between batch CVs. We've got an excellent monoclonal to PC2, and so we will integrate that into our schedule as well. We'll develop a FACS assay where we can look at individual exosomes from undiluted urine and measure the ratio of PC1, PC2 to CD133 per exosome. We'll integrate Cu62 into the West and perhaps a FACS biomarker assay. And this assay is actually being used by Regulus Therapeutics right now in their clinical trial. And tomorrow, Dr. Tanya Valencia will be talking about that. So these are the people that did all the work. Myself, Chris Ward, Wendy Lee, Madhulika Sharma, Kerry McGreal, Lessa Zelenchuk. And not on this slide, but equally important, Tanya Valencia and Edmund Lee from Regulus Therapeutics. So that brings me to the end of this talk, and basically any questions.